So I'd like to welcome everyone, and before I introduce Scott, I want to mention that Scott's lecture both today and then there'll be one tomorrow at 4:10 in 100 Hunt are being supported by an endowment established by Ruth and Tracy that has enabled us to bring in outstanding speakers in the biological sciences for a very long time, and we're very grateful. And Scott certainly is someone who merits being brought in as one of the most distinguished biologists today. I could spend a long time giving his background, but he got his undergraduate degree at Harvard, a PhD from UC Berkeley, and then was briefly at the University of Florida as a postdoc, and then was a professor at University of Washington, and in 2003 moved to Harvard to his current position. And he's gotten all sorts of honors, ranging from election to the National Academy of Sciences just a couple of years ago, a fellow of the American Association for the Advancement of Science and the American Academy of Arts and Sciences, and has broad interests across ornithology and evolution, and is also a really superb public citizen. Someone with a background like this then recently spent a couple of years at NSF as division director in biological infrastructure, where I got to interact with a bit. And Scott keeps pointing out that he kindly contributed an article to a encyclopedia I did on the Encyclopedia of Theoretical Ecology, so that shows how broad he is. And I was just remembering that a few years ago, you don't know this, I was in Paris and saw there was this wonderful public lecture being advertised, and it was Scott Edwards talking, and unfortunately I didn't get a chance to go then, but maybe we'll hear something today. I was wondering, did you do it in French or English? And the title's up there, so without any more, let's welcome Scott.
Thank you very much, Alan. It's great to be back in Davis. It's been so much fun today, seeing old friends and meeting some new ones. I just want to thank Alan again for the generous invitation, and Carol Holm and her colleagues for setting up my visit here. It's been a really extraordinary day. And yeah, as you can see from the sort of montage here, we're going to sort of take a journey through dinosaur evolution, avian evolution, throw a little bit of DNA in there. I've been fascinated by birds since I was a kid, probably 8 or 9 years old. I grew up in Riverdale in the Bronx, which despite being in the Bronx actually had some trees, and was really, back in the 70s, was overlooked the Hudson. It was a really wonderful place to grow up. And when you first, you know how when you go to a new place and you see these really interesting looking birds in the field guide, and you sort of say to yourself, oh, this one is so cool looking, I'm never going to see it. That, for me, that bird was the northern flicker. Yellow-shafted flicker, red-shafted flicker, and lo and behold, when I actually saw one, in my backyard, that was it. There was no turning back. So I want to develop this idea that birds are dinosaurs, and that we can understand their ancestry not only by looking at their visible traits, but at their DNA. So we're going to begin with sort of a broad brush intro to bring you up to speed on some of the latest thinking about the dinosaur ancestry of birds. And I want to tell you a little bit about why bird genomes and chromosomes are so interesting. They turn out to be sort of minimalist toolkits, much smaller, for example, than the human genome. And yet we know that birds manage to do incredible things with just about 1/3 the amount of DNA. We'll talk about some of the innovations that birds have come up with, things like how feathers evolved, and some interesting cases of evolution tinkering with what's available. We'll talk, for example, about how some birds, such as hummingbirds, have an ability to taste sugar despite the fact that they don't have the gene that makes the protein for tasting sugar. So how do they do that? And at the end, we'll discuss a theme that's been important in evolutionary biology, namely, what are the important drivers of evolutionary change? On the one hand, we can think about genes changing their sequence over time, evolving new functions. But we can also imagine a scenario where the regulators of those genes, the ones that are the switches that are telling genes when to turn on and where to turn on, maybe those are the important drivers in evolution. And that's it's been quite an interesting and lively controversy over the years. Briefly to tell you where I'm from, we're very fortunate at Harvard to have a vibrant museum, the Museum of Comparative Zoology. This is the old building in brick where the ornithology collection that I head has been housed for the last 150 years. In the last 2 years, it's moved to the basement of this modern building that you see in the lower left. Thankfully, our dean saw fit to keep it on campus. Collections take up a lot of space, but we try to convince them that they're exceptional resources for research and teaching. Through collections, we can really travel the world, look at variation among different species, different times, different places, and increasingly museums are important archives for genetic resources. You see there on the lower right this liquid nitrogen cryo container where museums around the world are continuing to collect genetic samples so we can archive genetic change as our environment changes. And I like to think of specimens as kind of a snapshot of a particular environment in a particular time. The amazing tools that can be brought to bear to unravel the mysteries in specimens like these Hawaiian honeycreepers, using the tools of chemistry, of physics even, are really, it's bringing new life to centuries-old collections. Now, the founder of our Augusta Museum was this man, Louis Agassiz. He was an interesting character, very famous for his theories on glaciers, and albeit somewhat of a racist, quite frankly. So he'd probably be appalled to know that I was leading the bird department. But you can see here him standing in front of this chalkboard, which when we moved the ornithology collections just a few years ago, we actually uncovered for the first time in over a century. And so me being the prankster that I am, couldn't resist having a little bit of fun. You can see me here trying to recapture the august stare of Louis Agassiz there. I'm not much good at invertebrates, but I try. Now, as with most stories in evolution, our story begins with Charles Darwin, and many of you will recognize this image from The Origin of Species as the only image to occur in that book published in 1859, same year as the Museum of Comparative Zoology, and showing a hypothetical genealogical tree, one in which the ancestors and lines of descent are purely hypothetical, showing extinction, some successful lineages. If you have ever been to Down House south of London, you will have appreciated just what an amazing scholar Darwin was. You can visit his study and see his original copy of Marx's Das Kapital. Up on the second floor there are evolutionary themed games for kids. My kids were quite young when we visited. Things like air hockey continental drift, where the continents are moving around. Now, of course, as an evolutionary biologist, this was like going to Mecca. This is the epicenter, and in a very real sense, evolutionary biologists are still testing Darwin's theories. And so here is me several years ago now, deep in thought, tracing Darwin's path around the sandwalk, where he would tend to walk at about mid-morning after a few hours of writing. Now, about a few years after The Origin of Species was published, first this remarkable feather was discovered in limestone quarries in Solnhofen, Germany. and then a few years later, this remarkable specimen of Archaeopteryx. And this was in many ways a boon for Darwin because, as many of you will remember, one of his biggest conundrums was the lack of intermediate forms in the fossil record. How can we explain macroevolutionary patterns when we don't have these intermediate steps? Archaeopteryx fit this bill perfectly because, as this diagram will show, it's an amazing hybrid of reptilian features on the one hand and avian features on the other. Archaeopteryx had a very long bony tail, which modern birds don't possess. It had these unfused digits on the forearm, again, very much an ancestral condition. And of course, it had teeth, which modern birds have lost. Still, as you can clearly see in this fossil, the feather impressions were substantial, and we now know that many Archaeopteryx specimens had substantial feather coverings on the hind limbs as well. And it shows a number of very sort of gracile features of the skeleton, showing that it was much more, its skeleton was much more lighter and possibly even pneumatic, showing that it evolved much more, at quite an advanced stage relative to its reptile ancestors. Oops. Archaeopteryx didn't have the large bony keel that we see on modern birds, although it did have a very diminutive clavicle or wishbone, which you can still see on modern birds such as turkeys. And so it was really up to this gentleman here, Darwin's bulldog, Thomas Henry Huxley, to propose for the first time very firmly that birds were likely descendants of dinosaurs. Huxley was, as many of you know, the premier comparative anatomist of his day, and he was very, he really saw ahead into the future as he looked at fossils such as this Compsognathus. Compsognathus is a turkey-sized dinosaur that was one of the earliest dinosaurs discovered, and saw the sort of obvious superficial similarities between the long hind limbs, for example, of Compsognathus and those of birds or other sorts of, or even crocodilians, which you see here on the far right. And so it was, without going into too much detail about the history of dinosaur origins of birds, it's been a theory that has sort of waxed and waned over the centuries. And I would argue that 99% of paleontologists and evolutionary biologists are now comfortable with the statement that birds are living dinosaurs, or at least the living descendants of a particular lineage of dinosaurs known as theropod dinosaurs. It's not universally held, but it's very much the number of fossils that have now come to bear on this link is really quite extraordinary. Many of them having been discovered in China over the past 20 or 25 years. And so now we have a remarkably detailed view of the transition from crocodilian ancestors. Crocodilians are the closest living relatives of modern birds, although there might not be much in their anatomy which suggests being a bird ancestor. But from the fossil record, we have an extraordinary series of fossils which clearly lay out many of the major transitions which converted quadrupedal lizard-like ancestors into the bipedal flying descendants that birds are today. And so we can make use of this fossil record and try to understand what are some of the drivers of these amazing innovations. One of the most interesting innovations of birds, in my opinion, is the respiratory system. Unlike mammals, which have a diaphragm and which expand their lungs by drawing air in through the trachea and out the same way, birds have a remarkable series of what are called air sacs that not only infiltrate the major body cavity, but in fact infiltrate many of the long bones and even the vertebrae of the skeleton. And graduate student paleontologist about 10 years ago now, Leon Claessens, reported for the first time that the incredible pneumatization, sort of the many cavities in which these air sacs follow in the skeleton, were found in many so-called non-avian dinosaurs, dinosaurs that clearly didn't have many of the other adaptations for bird life. So you can see a lot of these tiny little holes that Claessens identified are hypothesized to have harbored these little alleyways that the air sacs form, basically turning the entire skeleton into a big breathing apparatus. The other remarkable thing, of course, is that birds and, by inference, dinosaur ancestors have a very rigid lung. So the lung does not expand or contract. Rather, the air flows in one direction from the caudal end, the tail end, towards the cranial end, the head end. So it's a unidirectional airflow through the lung. And in fact, this unidirectional airflow has been discovered in crocodilians as well as some other reptiles. So it's a very ancient adaptation, but one that's thought to be quite a bit more efficient than the mammalian system. Now, this very efficient respiratory system has its own imprint on the genome itself. And much of my research in the past several years has been looking at this interesting question of how streamlined is the avian genome, and is the avian genome streamlined in the same way that its anatomy and physiology might be streamlined. So if you look at a typical human gene, of course we know that genes are composed of exons, the actual fragments that encode the proteins, as well as these so-called introns, which are sort of these intervening sequences whose function is still relatively unclear. It's quite striking that a typical human gene is vastly bigger than the corresponding gene of a chicken, which you can see here at the bottom of the slide. They both encode the same information. These colored boxes are the exons, which ultimately pass their information on into the mature protein. But it's these introns which are so drastically different between mammals and birds. And so the hypothesis was suggested by Austin Hughes in a really interesting paper in Nature that bird genes and bird genomes might be small in part to facilitate the high energy demands related to flight. So Austin Hughes took about 100 chicken genes, found the 100 homologs of human genes, compared their introns and say, wow, chicken introns are so much smaller. They've probably lost a lot of DNA because of flight. Now, how is this connection made? The logic was as follows. The logic is that to facilitate the high energy demands of flight, it'd be helpful if we shrunk bird cells so as to increase the sort of surface area to volume ratio. Once you've shrunk bird cells, it's not a long leap of intuition to suggest that the genomes themselves might shrink as well. In fact, it's a well-known correlation between cell volume and genome size that's actually well established across many different animal groups, plant groups, many different tissue types, etc. What's perhaps a little less clear is the extent to which small cells actually facilitate higher metabolic rates. The other question, of course, is to what extent we could believe in this connection between small genome size and flight when we're only comparing two species. Evolutionary biologists know quite well that a two-species comparison is simply not enough to rule out the many other factors that could be contributing to the small genomes of birds. So the question remains, why is this? Why do birds have such small, streamlined genomes? Well, to give you a broader view, we can look at this chart showing the variation in total genome size for various groups of amniotes, or egg-laying vertebrates. So we have the mammals, for example. Human genome clocks in at 3 billion base pairs, or gigabase pairs. Kangaroos have some slightly larger genomes, but in fact, things like tuataras, which are this ancient lineage of lizard that lives in New Zealand and likes to bask at 50 degrees Fahrenheit, it's a very strange lizard, have genomes quite a bit bigger than the human genome. Of course, are tuataras more complex evolutionarily? Well, perhaps not. This whole disconnect between genome size and phenotypic complexity is called the C-value paradox. And of course, many organisms like corn have genomes that are 10 times as big as the human genome. So it's unclear how those two connect. And here we have the birds at the far right with having genomes typically about 1/3 the size of the human genome. There's quite a bit of variation within the birds as well. So we looked at this question in a relatively old paper now, basically trying to ask, do we see a correlation between metabolic rate on the one hand and genome size on the other? Hughes's prediction would say that as metabolic rate increases, we should see a shrinkage in genome size. And so what we've done here is plotted the distribution of genome size on the one hand, on the left here, and metabolic rate, looking not only within the birds, but across the crocodilians as well, which that's what birds and crocodilians comprise the Archosaurian lineage, as well as squamate reptiles, mammals, and amphibians. Of course, the data set back then, 15 years ago, was vastly smaller than it is now. What was intriguing to us is first just how differently these two traits evolve. We could show, for example, that metabolic rate evolved in quite a gradual fashion. It was displaying what evolutionary biologists call Brownian motion behavior, where we don't see very drastic jumps between different levels of metabolic rate. By contrast, genome size shows quite a punctuated pattern, with, of course, many salamanders showing vastly larger genome sizes than their close relatives. We could find the predicted correlation or inverse correlation, at least when looking within the amniotes and within the archosaurs. But across vertebrates as a whole, it didn't seem to hold. Now, fast forward several years, we have the genomics revolution and a huge amount of data available on the online databases. And a graduate student at Harvard and I got together to ask the question, can we see evidence of a role for flight in the intron sizes of a variety of mammals, birds, and Anolis lizard? Can we see any predicted variation in intron sizes across these groups? And we were very surprised, or at least I was surprised, to find that both bats and birds show a predicted decrease in size relative to their non-flying sister groups. So the average intron size of bats, at least for the 500 or so genes that we were able to look at the time, showed detectably smaller introns than did their non-flying sister groups. So this was, for us, an interesting, compelling confirmation of Hughes's idea. Now, the story gets a little bit more complicated because, of course, in this study, we're dealing strictly with living species. We're still, although there were initial reports back in the glory days of PCR when people thought they were amplifying dinosaur DNA, and, of course, movies like Jurassic Park perhaps have convinced some of us that this is actually possible. It's never going to happen. And so we have to use, but at the same time, we might want to gain insight into what dinosaur genomes looked like and what were the ancestral genomes that ultimately gave rise to those of modern birds. So we actually looked at the paleo record to get a sense of, to try to generate some estimates of genome sizes for the ancestors of modern birds. This is where my colleague Chris Organ comes in. Chris received his PhD from Montana State University, and under the tutelage of none other than Jack Horner, the paleontologist depicted in Jurassic Park. So he was very well equipped to, on the one hand, try to establish a correlation between genome size and a cell size that we could actually measure in the fossil record, and then use that correlation to sort of predict or estimate the genome sizes of extinct lineages of extinct ancestors of birds. So Chris was able to identify a cell type, namely osteocytes, which are the sort of lacunae, the gaps left in the bone matrix once the living tissue has dissolved away, that could be used as a proxy for cell volume. And if we can establish a relationship between those cell volumes and genome sizes of living species, we can then extrapolate that to extinct species. So you can see from some of this data, these are two cross-sections of long bones from a theropod dinosaur on the left, known as Deinonychus, who was described by John Ostrom at Yale University in the 1960s, and a mammalian species, brown bear, on the right. And you can see with your own eyes, these are both micrographs at the same magnification, that the osteocytes are detectably larger in the bear versus the theropod dinosaur. So you can probably see now where I'm going with this. After compiling estimates of osteocyte volume from a variety of living and extinct species, we were able to lay out across a phylogenetic tree our predictions for and estimations for genome size in living species and ancestors. And so what you see here is a phylogenetic tree showing a range of living species whose tips end up at the present here, and extinct species as well. So we've got the variety of birds here, including living birds as well as ancestral theropod dinosaurs. We've got representatives of the other major branch of dinosaurs, namely the ornithischian dinosaurs, things like Triceratops. And we also have a number of living reptiles and living mammals. And the coloration is such that the very hot pink lineages are those which have genome sizes either known to be or estimated to be small and bird-like, versus those in the purple and blue, which are known or estimated to be much more on par with modern reptiles or mammals. And what's intriguing is that we were able to infer the possibility that small genomes arose far longer before the origin of flight itself. So if we plot the origin of flight, we peg it right about here where we see fossil species known from China, such as Confucius ornis, displaying some of the first feathers and first evidence of flight. But the actual first appearance of streamlined genomes we infer to have occurred several tens of millions of years earlier. So we can make the statement that flight probably wasn't the initial driver of small genome size in the avian lineage. It's probably driven by strictly metabolic demands. And in fact, the debate about whether theropod dinosaurs were warm-blooded, how active they were, I won't say it's been settled, but I think paleontologists are much more comfortable now with the thought that theropod ancestors of birds were highly active, of course very predatory, and fully consistent with a lifestyle that might be conducive to small cells and small genomes. So this was a nice way to sort of separate the appearance of flight from the origin of small genome size. Now, as you can imagine, part of the reason I put dinosaur in the title of my talk today is to get people in the room. People tend to stand up when they hear the word dinosaur. And naturally, a lot of the press was interested in our findings. And so we had some interesting discussions with reporters on this, one of whom pointed out that, well, the headline was that big dinosaurs had small genomes. And I was like, well, we didn't exactly say that. There are actually a lot of small dinosaurs that had small genomes too. And then somehow it came up with the conclusion that, but size doesn't imply inferiority, study says. I don't know where they got that from. But now, Ernst Mayr was one of the most famous evolutionary biologists of the 20th century. And he had a lot of opinions about sort of the future of evolutionary research, phylogenetic research, One of my favorite quotes was this one from his 1942 book, Systematics and the Origin of Species. He said, "The study of phylogenetic trees comprised a field which was the happy hunting ground of the speculative-minded taxonomists of bygone days." I think I'm what he's talking about there. I think he would be astonished to learn about the incredible advances that have been made in phylogenomics and the history of modern birds that have just appeared in the last few years. And so fast forward another 10 years after his passing, and we have this remarkable overview of the genealogy of modern birds informed by the sequences of entire genomes of about 50 species. This was the result of a large international consortium, which I and many others played a part. And the goal was to learn a bit about how the genomes of birds evolved and to help solidify, to sort of provide a framework for the evolutionary history of modern birds. And you can probably find your favorite lineages up there. For example, we know that the Paleognathous birds, the flightless birds, which you see here on the right, comprise the deepest branch of modern birds. And this is what I'll be talking about a bit tomorrow in the departmental lecture. On the other end of the tree, in green here, you can see some of the so-called vocal learners. These are birds that we know require a tutor to learn their song. And so the Oscine passerine birds are a group of about 4,000 small-bodied birds that live on virtually every continent. And they're classic examples of vocal learners. And in fact, one of the best models in biomedical science for vocal learning is the zebra finch, a small finch that lives in Australia that is amenable to experimentation and, like humans, requires a tutor to learn its song. We were also able to show that there's been some remarkable radiations. For example, this group here in blue comprises many species associated with water or marine environments, suggesting that many of these adaptations perhaps arose deep in the ancestry of this group. And so it was a nice sort of first shot across the bow in terms of sort of genome scale phylogeny of this amazingly rich group of vertebrates. Now there's one lineage here called the Hoatzin. It's Opisthocomus is the genus name. It's quite an enigmatic species, and it had essentially foiled and tripped up Systematus for well over a century. Now the dirty little secret is not even our whole genome data set and lots of sophisticated number crunching could definitively nail down the phylogenetic position of Hoatzins. And for whatever reason, Science magazine felt it appropriate to put the one species that we couldn't figure out on the cover of the magazine. Go figure. Now there's this sort of genealogical portrayal sort of raises again a lot of very interesting questions about broad patterns in avian evolution, in particular biogeography. And I just want to briefly, before returning to the genomic theme, I want to just mention a couple interesting themes that are sort of dueling for primacy right now in the field of avian biogeography. If one takes a phylogenetic perspective and one takes those trees like I showed you on the previous slide and tries to infer what the ancestral domains of many lineages of birds might be, one comes up with the conclusion, championed by people like Joel Craycraft today, that many modern groups of birds had their origins in the southern hemisphere, in Gondwana land. For example, we find that many of the sister groups, sort of the earliest branching group relative to another group, are found in the southern hemisphere. So things like the magpie goose in Australia, as well as megapodes, these are all sister groups either to all the other ducks and geese or to the remaining chicken-like birds. And so this sort of phylogenetic argument suggests a Gondwanan southern hemisphere origin. And it's really quite remarkable to see the scale on which phylogenetic analyses are occurring these days. This is just a recent paper from the literature showing a quite comprehensive tree, at least comprehensive in terms of the number of species it's sampled, well over 1,000 species. And you can see the coloration here with New World in green, Old World in brown. This is the entirety of the passerine lineages, and the passerines, of course, are the largest radiation of modern birds, comprising over 5,000 of the 10,000 species of modern birds, and using these broad-scale phylogenies to infer major routes of origin. And like the megapode and magpie goose I showed you on the previous slide, passerines themselves are thought to have a Gondwanan origin based on the pattern of biogeography inferred from phylogenetic trees. And this is just a depiction of one scenario published by Per Eriksson in Sweden showing a Gondwanan origin and a spread all across the continents. And so, for example, our crows and jays here in North America are very clearly had their ancestors in Australia, and they crossed over into North America via the Bering Strait, making their way all the way down into South America. Now, the contrasting perspective is coming not from phylogenetics, but from paleontology. And in particular, Gerald Meyer, a paleontologist in Germany, has been championing a northern hemisphere origin for many modern groups. And for his data, he refers to a remarkable array of fossils, many of them assignable to modern groups of birds that are very well preserved in northern hemisphere deposits, many of them in Europe. And so, for example, this remarkable fossil of a Eocene hummingbird found in France and Germany. As you well know, hummingbirds today are only found in the New World, okay? They don't occur in Europe and the Old World. And yet here we have fossils that clearly show that hummingbirds were present in the Old World at some point. So it's, I can't say that this debate is resolved, but it's something to keep an eye on in the upcoming years. Now, I want to spend the last part of my talk to talk about some of the ways in which genomicists such as myself are using the tools of modern genomics to understand the origin of avian traits. In particular, things like the origin of feathers, and what I'll be talking about tomorrow, the origin of flightlessness. And now that we have sort of access to all parts of the genome, we can begin to look at the sort of dark recesses of the genome to try to understand what's producing these remarkable adaptations. In particular, we can look at what's called the non-coding dark matter part of the genome. As many of you know, the amount of the genome that actually encodes a protein, whether it's a human or a bird, is only about 2%. So that leaves essentially 98% of the genome whose language and whose interpretation we still don't fully understand. And there are major projects ongoing trying to unlock the sort of language of that major part of the genome. One hypothesis is that the non-coding part of the genome forms a vast regulatory network that is tasked with as switches to turn on genes and turn them off at the appropriate times during development, what we call a regulatory toolkit. Now, the debate as to which is more important, gene regulation or actual changes in the genes themselves, has been going on for a number of decades. And here's a picture of Alan Wilson, whose lab at Berkeley I was very fortunate to work in as a graduate student, where he proposed, along with Mary Claire King, who's now a geneticist at the University of Washington, that the number of differences that could be found in the proteins when comparing humans on the one hand and chimps on the other, that number was so small that you had to explain the morphological and behavioral differences based on gene regulation. The differences in the proteins themselves were far too minor to explain the vast differences in morphology and behavior between these fairly closely related species. And so in some of our recent research, we've tried to tackle this question from various points of view. I want to first show you an example where it's pretty clear that there's a very tight connection between changes in the sequence of a gene and the origin of a new function. So the ability for animals to detect taste, to detect compounds using taste receptors is a really great model for looking at the relationship between genotype and phenotype. And we know that our taste receptors is a very small multi-gene family, essentially only 3 different genes that are expressed on receptors all across our tongue, and they essentially form heterodimers, one of which is able to detect umami or savory taste, essentially amino acids or the taste of meat, and another of which is targeted for sweet taste perception, sugars and carbohydrates. Now, there have been a number of natural experiments out there that have shown us in dramatic detail how these taste receptors work. So, for example, pandas, whose diet is basically 100% bamboo, have essentially jettisoned their umami receptors. They don't have them in their genome anymore because they're not used to detect anything meaningful for them. Cats, it turns out, although they love to sip at milk, have actually lost their sweet taste receptor. And so we have interesting examples from throughout vertebrates of how loss of a gene is coincident with the specialization of particular diets. Now, when it comes to birds, we've got a really interesting experiment to look at because we know that bird genomes today do not possess the gene that mammals used to detect sweet taste perception. That gene has essentially been lost from every bird genome that's been looked at. And this, of course, is consistent with the carnivorous origins of bird lineages. And so T. rex and its relatives were all meat eaters. They had no use for a sweet taste receptor. Archaeopteryx on through moas. And of course, modern birds, such as fairy wrens and hummingbirds, have inherited the genome of their dinosaurian ancestors. But how is it then that they're able to detect sweet compounds, such as nectar, when they don't have the gene encoding a sweet taste receptor? Sorry, this is in Spanish. It's from a talk I gave in South America last year. My graduate student, Maud Baldwin, wanted to attack this problem, and this was one of those moments, as I'm sure many of the faculty here appreciate, where your student is proposing this outlandish project that you suspect will never be completed, and you do everything in your power to dissuade them from going down that path. And like a good stubborn graduate student, she stuck with it with, I think, really spectacular results. So she was interested to figure out how it is that hummingbirds actually detect sweet compounds. And she knew that hummingbirds clearly can detect sugar. We know that from many behavioral experiments that have been done with hummingbird feeders, and that their sister group, the swifts, could not detect sugar, as could not galliforms and chickens. So we have an innovation occurring, and the question is, how do they do it? Well, she went to the nearest gene that she had a hunch might be involved, and that was not the sweet taste receptor, because they don't have it, but the umami receptor. And she was able to show that the umami receptor in birds has morphed through a series of amino acid substitutions, indicated by these stars here, into a functional sweet taste receptor. So here's an example where changes in the sequence of a gene and a protein have evolved a new function. So this was different from the vision of Alan Wilson and Mary Claire King, who believed that changes in gene regulation were really what's important in evolution. But let's look at another trait. Let's look at the origin of feathers, which in many ways are sort of the iconic avian trait. And we're going to get a handle on the sort of regulatory landscape of the genome by looking at these regions of the genome known as conserved non-coding elements, or conserved non-exonic elements. What this means, these are regions of the genome which show a high degree of conservation between species. They clearly haven't changed over evolutionary time. And that's indicated here by this very dark region, where you can see species from monkeys to mouse to other mammals, chickens, frogs, and fish. This region, which is not found in any gene or coding region, is highly conserved. Sequence is very similar. We know from population genetic theory that such regions likely have an important function. It's unlikely that something would retain its sequence for so long without serving an important role. We might not know what that function is, but we can bet that it's got an important function. And today, many biologists believe that these conserved non-exonic elements serve as what are called enhancers to help orchestrate the expression of genes, telling them when and where to turn on and off during development. Now, we wanted to look at the relative roles of protein coding genes on the one hand and regulatory innovation on the other hand in the origin of this iconic trait, namely feathers. We know from the fossil record a lot about when feathers evolved and likely for what purposes. We know that a lot of non-avian, non-flying dinosaurs had very robust feather coverings, which were likely used for sexual display or for thermoregulation. And scientists today will even tell you the colors sported by many extinct species using the microscopic structure of the melanosomes, these organelles that help modulate feather color. So how are we going to get at the molecular origins of feathers? Well, this was essentially an entirely bioinformatic exercise. And what we did was we compiled a series of genomes from not only birds, but from reptiles, mammals, amphibians, all the way down to fish. So we could look at the sort of deep origins of feathers at the DNA level. Then we scoured the scientific literature for candidate genes that developmental biologists had told us played a role in feather development. Okay, and we found almost 200 feather-related genes, including the beta-carotenes, which of course are the major sort of structural component of modern feathers. We used computational approaches to identify a very large number of these conserved non-exonic elements, these regulatory landmarks around the genome. About 13,000 of these elements were very closely linked to these 200 feather development genes, and so were likely involved with their regulation. And this exercise basically consisted of tracking the rise and fall of both the genes that are thought to be involved with feather development, as well as the regulatory elements. So we're dealing with some very complex landscapes here. Here's a snapshot of some aligned genomes where we see a gene called S-HOX. S-HOX is one of the 200 feather genes that we looked at. You can see that it's very conserved across vertebrates. You can see the exons are found all the way down to fish, in some cases all the way through the mammals and reptiles. And you can see that these conserved non-coding elements, these enhancers, are sort of sprinkled around the gene, and I've color-coded them here so you can see where in evolution they arose. So the red ones are bird-specific. They're found only in bird genomes and not in anything more ancestral, whereas this green one, for example, is found also in the Anolis lizard genome, and this black one, for example, has evidence for occurring all the way down to fish, so it's a vertebrate-wide element. And so by tabulating sort of thousands of these genes and elements, we can sort of track which one is sort of arising coincidentally with the origin of feathers themselves. Now, the first thing we looked at were the keratin genes. And in fact, keratin genes are well known to have undergone a major duplication in the lineage leading to modern birds. So that's a clear point in favor of protein evolution being important for feather origins. And we've tracked that here by, on the y-axis, we basically have a measure of the over or under representation of these elements at a particular slice of evolutionary time. On the x-axis, you can see it's sort of a timeline going from fish on the left all the way up to birds on the right. And so we can show here what we actually knew from other studies, namely that the beta-carotenes underwent a dramatic bout of gene duplication right in the lineage leading to modern birds. And so that was sort of confirmatory evidence. What for me was more interesting was to look at the non-keratin genes. So these are all the other developmental patterning genes that help create the feather, placode, feather bud, and the mature feather. And here we see a very different picture where many of the genes that are involved with feather development, indicated here in green, are actually quite old. Just like you saw with the S-Hox example in the previous slide. We can find many of the genes that are making a feather in fish and amphibians, even in humans. It sort of inspires me to just spread my wings. I've got essentially 100% of the toolkit, of the genic toolkit to make a modern feather. What humans and non-avian vertebrates don't have, however, is the full regulatory toolkit, as you can see in this blue line here. What you see here is some evidence that the rate of origination of these regulatory elements increased quite dramatically, actually not in the ancestors of modern birds, but in the ancestors of amniotes. Remember, these are the egg-laying vertebrates. This regulatory explosion seems to have occurred as vertebrates moved from water onto land, sort of in a very gross sense, moving from amphibians into lizards and mammals. So there's quite a bout of regulatory innovation around these feather genes when amniotes moved onto land. But that innovation continued on all the way on through the reptiles and through the birds. So we suspect, in fact, that while the coding regions that help put a feather together are quite old, and many vertebrates possess nearly the entire toolkit, it's the regulatory innovations which appear to have arisen more coincidentally with feathers themselves. And in fact, we see a big signal for a repatterning of these feather genes as vertebrates moved on to land. Now, we were also, just as sort of how am I doing for time? Am I okay? Or we started a few more minutes. Okay, good. We were also interested in asking, from a genomic standpoint, what sort of makes a bird a bird? Where in the genome do we see a lot of the bird-specific innovations occurring? And so we scanned the genome for regions that contained a very large number of these regulatory elements, specifically those that arose on the bird lineage. And that's what you see on this graph here. Here you have number of elements per megabase, and here we're taking a scan across the 7 major chromosomes of chickens. And we actually found one region here on chromosome 7 that had a density of these bird-specific regulatory elements that was over 500 times the genome-wide average. What's intriguing is that these regulatory elements are quite close to 2 genes called insulin growth factor binding proteins, which are well known to play roles in body size change and even changes in limb proportions based on studies of domesticated chickens. And we suspect, in fact, that regulatory change in and around these genes was responsible for this amazing trend in body size evolution that we see in the lineage leading to modern birds from dinosaur ancestors. And so in a very interesting paper published by Mike Lee, a paleontologist in Australia a few years ago, he showed that one of the major trends in avian evolution was the miniaturization of body size occurring over tens of millions of years. We hypothesized that these insulin growth factor binding proteins were involved with that process. And this, I think, is actually a testable hypothesis with modern biotechnology approaches. So hopefully I've given you a sense of some of the interesting origins of the avian genome, some of the ways in which genomicists can look at the evolution of avian traits. And I hope you're able to attend my talk tomorrow, where I'll be applying some of this logic to study the evolution of flightlessness in these Paleognathus birds and ratites. And I'd just like to thank my lab group for contributing to many of these studies. Thank you very much.
So the story on the hummingbird sweet receptor makes me wonder if anyone's done behavior to see if they have an attenuated sensitivity to umami. Because they have a niche where both sugar and protein is important for them.
That's right, yes. And I don't know if Kurt Klasing is here. He was very instrumental in this study as well. In fact, Maud came to Davis for several months to get this project going. I think it's entirely possible. We know that hummingbirds eat a lot of mosquitoes and whatnot. They're not strictly nectar feeders. And Maud has some preliminary evidence that this receptor, this sort of heterodimer, can in fact bind some amino acids as well. So it's not completely 100% specialized on carbohydrates. So yeah, that's work to be completed, but I think it's likely a more general receptor than we originally predicted. Yeah. Yeah.
So why do you think the birds made it through the KT boundary and the dinosaurs didn't? And also, why did the toothed birds not make it through, but the toothless birds did?
Well, let's qualify that statement a bit. So, of course, since birds are dinosaurs actually did make it through the KT boundary. I mean, I know it's kind of semantics, but that's the way we think of it now. Yeah, and there were some toothed birds that actually survived the KT. I mean, things like Hesperornis and Ichthyornis. I think, the role of the KT both in bird evolution and mammal evolution is uncertain now. I think there's not a lot of evidence from phylogenies that this cataclysm actually sort of shrunk the phylogenetic tree to just a few surviving lineages. I mean, people have tried to look at sort of the diversification, how diversity accumulates through time in both birds and mammals, and it's actually challenging to find a strong blip when you would expect it for the KT boundary. So, I think surviving lineages probably were living in niches that were somewhat robust. I mean, mammals at the time many of them were very small-bodied, nocturnal, and living in burrows. But we don't see a super strong evidence of mass extinction, at least when looking at molecular phylogenies.
I wonder if there's any correlation, say, or any evidence or anything of, say, if you have fewer genes, if you need more regulatory underpinnings to get functionality out of it. So if we were to just, throw some more genes in corn or something, I mean, not that corn's lacking, but just that maybe for a plant then would you need less regulatory? Could you strip its gene again and get the same result better?
Well, that's a good question. I mean, somehow birds are able to do it with, 1/3 the amount of non-coding DNA as mammals. And, I'd like to think that they were sort of more efficient in terms of their regulation than mammals are. Of course, we know that, the human genome and other mammal genomes there is, in fact, a fair bit of junk in there. A lot of it's descended from transposable elements. But you see these really interesting examples where what was once junk has now been co-opted to do something important. And so I think it's a very interesting question, the trade-offs, investment in genes versus investment in gene regulation. And there's a guy named John Maddock in Australia who's thought a lot about this, and he actually thinks that it's really regulatory complexity that scales with phenotypic complexity. It's not the number of genes so much. It's more the regulatory toolkit when you look over sort of macroevolution. So, yep. Yep. Hey, Gino, how you doing?
Good.
Good to see you.
So you were talking about the two different ideas for evolution. Which do you think is more important functionally in terms of changes, the regulatory evolution or the protein coding? You give examples of both.
Good question. I mean, personally, and I'm going to perhaps contradict my graduate student, Hopi Hoekstra, who's more on the protein side, but I think regulatory evolution is going to be, in the majority of cases, what's behind many of the phenotypic diversity that we see. The truth is, I think evolutionary biologists have tried to tackle really the low-hanging fruit, some of the very simple phenotypes that may well have a basis in protein change. But all those other, phenotypes are complex. There's continuous variation, there's height variation, there's lots of things to quantify, and I think in a lot of those sorts of quantitative kinds of variation, it's going to be a lot of gene regulation. So.